Okay, so this is the last half day of the end game. We're going to have two sessions this morning. The first one, session 12, is building a path forward. What is the dengue endgame and how do we get there? We're lucky to have Dr. Jenny Loh, Dr. Andre Sequeira, and Dr. In-Kyu Yoon. This is a really, really important session because, as we've talked about a lot, it's all about implementation, right? It's all about taking everything, all this hard work that everyone puts in and all these discussions and figuring out how to way to actually apply them and make a difference. So we're first going to hear from Dr. Jenny Loh from Duke-NUS. The floor is yours.
Thank you, Stephen. All right. Good morning, everyone. And once again, thank you, Stephen, Adam, for the kind invitation for me to speak here in this summit. I'm very happy to be here. This is my first end game. So I was asked to talk on challenges of running clinical trials, maybe specifically therapeutics, not vaccines. And I titled my talk, Reimagining Dengue Clinical Trials. Just look at the decoration out of the way. And this slide, I don't think I need to go through it again. I'm speaking to the converted here. Okay, and I think we can also all agree that we need, there's no single countermeasure that will fully control the problem of dengue in the years to come. And as and when a therapeutic becomes available, it will form part of this important overall countermeasures to really control dengue and hopefully one day maybe eliminate dengue. And potentially, as it was theoretically, right, when there is a drug that can treat dengue, this drug should, in theory, reduce disease duration, symptoms, and as well as severity of illness, and reduce the overall short and long-term complications of dengue fever. Because you've also heard earlier on, I think in the first day, that there are long-term sequelae of dengue. And but as you can see from here, we have been repurposing drugs for dengue indication for the last 15 years. Various drugs have been tried, but we have not been successful. And more recently, the last 2-3 years, there are two novel compounds that unfortunately have also met with very early demise in the clinical stage of drug development. ATL752 is an NS5 inhibitor developed by ATL, and just after 21 patients recruited in the phase 2 study, the trial was terminated because the sponsor felt that the escalating cost and the long duration to recruit patients did not justify continuing the trial, the drug development, unfortunately. And of course, J&J in 2024 also decided to can both their prophylactic and therapeutic clinical trial in 2024 because the company has decided to reprioritize their, I suppose they have other priorities. Yeah. So that leaves us with just really a handful of drugs that are currently in the clinical pipeline. I'm not talking about drugs that are being developed in the preclinical stage. So you heard from Stephanie yesterday, EYU 688 is currently in phase 2 clinical trials, recruiting over 17 sites, including 2 sites in Singapore. That's in phase 2, and you also heard from Bert from Island Pharmaceuticals, that is the fenretinide, which is a repurposed drug, and just finished a phase 2 human challenge here in SUNY. And of course, VIS-513, this is a Serum Institute's monoclonal antibody pan-serotype monoclonal antibody against dengue, previously known as VIS-513, is now called dengue map. And we just finished a phase 2. In fact, the phase 3 recruitment has just started. I also understand that there'll be several more sites opening up, although currently is recruiting in India. And Sophie Jacob from Oxford also had recently been awarded a grant to start a new platform trial. She will first start by testing 2 repurposed drugs, again, 2 repurposed drugs that you can see up there, and with possibly adding VIS-513 to the list of drugs to be tested. But as far as I know, the platform trial has not started recruiting, and for the start, I think that it's only restricted to 1 site in Vietnam. All right, I'm going to skip this in the interest of time. So we know this is the conventional drug development timeline from preclinical all the way to licensure typically takes can take up to at least 20 years. And for dengue therapeutic drug development, specifically novel compounds, there are issues with preclinical drug development, which is not the focus of my talk today. There are obviously some limitations with how preclinical drug development is typically being conducted, but I'm not going to focus on that today. And for all the drugs, whether it's novel or repurposed, none of them actually have been terminated in phase 1 because of safety reasons. So all the drugs have been able to move smoothly from phase 1 through to phase 2, but all the drugs have been either failed to move on to phase 3 or prematurely terminated in phase 2. And so I think the rest of the talk, I really want to focus on how we can get around this problem of drugs failing to move beyond phase 2. Is it a problem really with the drugs, or is it a problem with the way we carry out phase 2 clinical trials when we test therapeutics against dengue? So these are all the failed drugs from previously, and if you can look at the primary endpoints, conventionally we have always stuck to 2 endpoints, a virological endpoint. In this case, we always measure RNA-emia for the convenience and ease of measurement, as well as a clinical endpoint of fever, because some of these studies are conducted in inpatient setting, which then allows the investigators to really monitor the temperature very, very closely at close intervals. And there's a reason why we have kind of stuck to these endpoints, because many, many years ago, we decided that in order for clinical trials to be comparable from study to study, we should harmonize endpoints. So these endpoints were chosen not for very valid reasons, right? Because from previous talks, again, we know that the difference in viremia actually does make a difference to the severity of illness. A 1 log difference can actually previously, people have shown, others have shown that a reduction in 1 log viremia can reduce the severity of dengue. And recently, the Vietnam group, from their cohort of 3 studies, I think combining data from 3 different cohorts of more than 2,000 children and adolescents, you can see from the graph on the bottom graph on your right that the rate of decline of the viremia will also impact the severity of the illness. The faster the decline, the less severe the clinical outcome. But we also know that the viremia kinetics vary very widely from individuals to individuals, as you can appreciate from the diagram on your left. And again, from the Vaughan et al. Paper, you can appreciate that after the first 48 hours, there's a very rapid decline in the RNA-emia. And it took about more than 2,000 patients' data points to get a very nice graph like this, very nice fitted graph like this. But in the phase 2 clinical trial, we typically only recruit no more than 200 to 300 individuals. So it is almost impossible to replicate the same kind of viral kinetics in a phase 2 clinical trial. And then we use fever as a clinical endpoint in phase 2 because you heard from Daniel yesterday, right, and Bridget yesterday, severe dengue is a very rare occurrence, so there's no way we can use any of those indicators as clinical endpoint in phase 2 clinical trial. So we have always said fever could be something meaningful to measure because, at least for the patient subjectively, right, as fever subsides, it is a meaningful endpoint from the patient's perspective. But fever also tracks very closely with viremia. So these are the fever curves from 2 previous trials, salgosvir and ivermectin. And of course, for those trials, there was no difference in the viremia the curve separating the treated and the placebo group. And similarly, when you look at the fever graphs, there is absolutely no separation at all. But so is it true that these endpoints are not useful at all? I think we can take a step back and maybe look at the Serum Institute's phase 2 trial findings. So this, I got this slide from Guek, very grateful to him for lending me the table, because the manuscript is actually not under revision currently, and it's not out in the press yet, but he's agreed to share the table with me. So Serum Institute's monoclonal antibody is a slow infusion monoclonal antibody, and in phase 2 they recruited about 250 patients and randomized them into 5 arms. There's a placebo arm, and then 4 groups of patients were then randomized to receive different doses of the monoclonal antibody given as a slow bolus infusion. And as you can appreciate from the table, there are about 50 patients in each arm, but only 20% of patients actually have fever as an evaluable endpoint out of the 50 patients. Yet, they can see a difference as the higher the concentration of the monoclonal antibody goes up. You can see that there is a appreciable difference in terms of median time to fever clearance, as well as the proportion of patients who experience, who became afebrile after 24 hours of receiving the monoclonal antibodies. So it really, I think at the end of the day, it is whether we are using the right endpoint for the right drugs. So I'm just going to quickly go through this study that we did a few years ago now. The results have been published. I'm not going to go into the details of the trial. Essentially, this was a novel monoclonal antibody against yellow fever virus that we brought to the clinics. In a phase 1 adaptive trial design, we built in a safety and dose escalation study with a proof of concept efficacy in using the YF17D vaccine as a challenge, because we know that this YF17D vaccine strain can induce viremia as well as cause symptoms in the vaccinees. And if you look at the graph on your right, for the efficacy stage, what we did was we recruited 10 healthy volunteers and randomized them to receive placebo or IV infusion of the yellow fever monoclonal antibody over 30 minutes. So we challenged them first with the YF17D inoculation, and 24 hours later, 5 subjects went on to receive the placebo, which was normal saline, and another 5 subjects received the yellow fever monoclonal antibody. And you can see here in the placebo group that 4 out of 5 individuals developed detectable YF17D RNA emia, whereas in the treated group, none of them became viremic after the infusion of the antibodies. We also tracked the symptoms that these subjects developed, as well as took the bloods to look at the gene expressions. And if you just look at the bottom of the diagram, these are the numbers represents the number of symptoms each individual subject reported after the injection. And for the placebo group, not surprisingly, many of them, 4 out of 5 did develop systemic symptoms. The numbers represented the number of symptoms that each individual reported. And in the treated group, only 1 individual reported 1 systemic symptom. But yet, this is not statistically significant because the sample size was very small. If we look at the molecular signatures of these individuals from infection from the time of pre-infection and post-infection, you can appreciate that actually in terms of the host molecular signatures, it is like day and night between the 2 groups. I'm not going to go through the immune genes that were upregulated in the placebo group compared to the treated group. I just want to show that the molecular signatures actually, because it's more objective, more sensitive, you can technically get away with a much smaller sample size if we decide to use molecular endpoints for the evaluation if we decide to use molecular signatures for the evaluation of endpoints in clinical trials. And because these changes in the gene expressions are actually significant between the 2 groups. Okay, this is not meant to be read because it's font size 6. This is to make the point that, and Stephen will appreciate this, because in phase 2 clinical trials, we typically only recruit very well individuals into the trial, except for the condition, the indication of the condition that we are testing, right? So we will not be, we screen them for a whole lot of exclusion, and what we recruit, the kind of patients we recruit into phase 2 are usually very, very fit individuals. But we know that the dengue disease pathogenesis is also driven by the host factors. And Richard yesterday said that, really severe disease is a rare event. We know as the viral load comes down, fever subsides, and then the inflammation-driven vasculopathy, sometimes during the critical phase, can become exacerbated, and it can then result in severe plasma leakage, hypotension, multi-organ failure, and this is a time when the clinicians will really need to treat the patient very, very carefully with fluid replacement, otherwise the mortality is as high as 30%. But most patients don't develop these complications, right? They get they have subclinical disease, or they get mild dengue fever. So then really, who are we looking for? Who are the ones that will go on to develop severe disease? We know from observational studies, from retrospective reviews, that older adults, diabetes, hypertension, chronic renal disease are all risk factors associated with severe disease, but we cannot recruit these people in the phase 2 trials because that will cause there's too much risk to bring these patients into an early phase clinical trial. And also, we also do not want to do that because then you introduce a lot of heterogeneity into the study population, which will make the trial findings extremely hard to interpret too. But we know in recent years that obesity on its own without associated comorbidities are increasingly being recognized as a risk factor for severe dengue, both in pediatric and also in adult population. And recently, Eva's group in Nicaragua, from their children cohort, also reported that actually obesity in children increased the risk of dengue infection and symptomatic disease. And we're not even talking about severe dengue here. Obesity itself predisposed these children to dengue infection. So in the interest of time, I'm going to skip over some of these slides. I'm going to go through data from a study that we did just quite recently, a few years ago, and the manuscript is currently under review. We had previously shown using the YF17D as a challenge model to really understand host response to an acute autoflaviviral infection. And because YF17D is a very safe vaccine, the side effects and the symptoms, as well as the immunogenicity, are quite well known. So we've been using it to like a challenge strain, so to speak. And we saw that the pre-infection ER stress actually predisposed individuals to more systemic symptoms. And we then asked whether obese individuals are at higher risk for developing systemic symptoms following an acute viral infection. So in this study, we recruited 35 individuals who have a high BMI. We define high BMI as more than 25. I know it's a bit low for some people, but using the Asian cutoff, we take 25. Then normal weight in so normal weight individuals are defined as those with BMI from 18 to less than just below 25. And we inoculated these healthy individuals with YF17D again, and then tracked the viremia as well as the host response. You can see the bloods that we collected from all these volunteers. And indeed, we confirmed that our hypothesis, if you look at the spider plot, the pink line represents systemic symptoms. Higher BMI individuals do have more symptoms, especially body aches, and they're also more likely to develop the lymph node swelling in the axilla on the same site where we gave the injection, and that's statistically significant. But if you look at the YF17D RNA-emia in the two groups, there is no difference, whether you calculate by area under the curve or we look at in a proportion of individuals who were detected positive for YF-RNA-emia on the days that on the days where they were sampled. There was no difference in the proportion as well. And we also looked at their actually we looked at a whole lot of things. I'm only showing you the cytokine data here because just to make up my point. It's not surprising that in individuals with higher BMI, we do see higher pro-inflammatory markers, and I'm just showing two here, IL-6 and TNF, that are both elevated in the obese, higher BMI individuals compared to the normal weight, normal BMI individuals. But more interesting is that the concentration of the cytokines actually track with the YF17D RNA EMIR, and the magnitude of the increase in the concentration is actually greater in those with higher BMI compared to those with normal BMI. And if we split the BMI into if we split those with high BMI now into quintiles, you can appreciate that the cytokines concentrations actually change with actually higher as the BMI goes up. Okay, so how to reimagine dengue clinical trial, right, is a question for this session. And we have heard again and again from various speakers over the last 2 days that dengue is a very complex disease. So the viral host interaction is very complex and dynamic. Viral kinetics change with days. The host response also is very dynamic with changes from day to day. And how these changes then interact when we give a therapeutic to the patient, how does a drug interact with the virus, how does a drug interact with the host, and vice versa, I think is actually quite a complicated thing when I try to sit down and think through this whole thing. But so I think we are boxing ourselves in when we try to say that, we need to fix the criteria and have everybody use the same endpoints. I think we need to be thinking about whether we have to change the entire concept and start thinking about whether we should be thinking more in terms of the mechanism of the drug action and the type of patients that we recruit. And so maybe there's really no one-size-fits-all endpoints at the end of the day. I think, how to solve the problem, right? I'm running out of time, Stephen, I'm aware, sorry. So I think we are now living in the era of the molecular humans. We now have a lot of technologies available within our reach. Omics technology, we can now gain really novel insights into the host immune response. We can measure gene expressions, proteins, metabolites, activities of the immune cells right down to single cell level, right? So how can we then use these tools and build it into clinical trials to help us advance therapeutics? This is just something I thought about. It hasn't been pressure tested yet. So I'm thinking perhaps instead of going through the conventional phase 1, phase 2, phase 3 kind of design, maybe we should be taking one step back now and say that, okay, once the drug has passed safety and tolerability in healthy volunteers, instead of going straight into a phase 2 clinical trial design in a randomized controlled trial with a few hundred patients, we should take one step back and maybe just recruit a single arm and do a single arm study, recruit maybe 30 to 50 dengue patients, put them through the drug therapy, and then using the tools we have really deeply characterize the PK/PD of the drug, as well as do deep profiling of these individuals using all the omics technology that we have, and then couple that with AI, ML, and machine learning, which we heard from Kerr yesterday, then perhaps we can then find the marker or markers that will really inform on what our endpoint should be when we move to a phase 2 clinical trial. So I'm just going to end here, and sorry for running over time, Stephen, and I'll be happy to take questions later.